Good morning, everyone. I'll just take a minute to introduce Professor Partha Majumder. Professor Partha Majumder is the current president of the Indian Academy of Sciences. Professor Partha is a fellow of all three Indian National Science Academies, the Indian National Co-Coordinator of the International Cancer Genome Consortium, and a member of numerous international scientific societies and editorial boards. He is the founding director of the National Institute of Biomedical Genomics and a professor at the Indian Statistical Institute in West Bengal. He is the recipient of several prestigious national and international awards, such as the G.D. Birla Award for Scientific Research, Ranbaxy Research Award in Applied Medical Sciences, and the New Millennium Science Medal. His major research interests include human evolution, and the genetic architecture behind disease susceptibility and drug response. Professor Partha has several more awards to his credit, but because we're short on time, I won't be able to read all of them. His topic for today is conducting human genetic research in India, then and now, some personal reflections. Welcome, sir. The floor is now yours.
Thank you very much. It's really a pleasure to be here this morning and to give you some personal reflections on the development of human genetic research in India. I've titled my talk as I've left out a word, as you can see from the slide. I've left out a word called conducting. I've just called it now human genetic research in India, then and now. And what I'm going to do is primarily focus on the then part and play down on the now part, primarily because most of us are in the business of conducting human genetic research now. I'm giving this talk to the membership of the Indian Society of Human Genetics, and therefore all of us are in the business of conducting human genetic research now. So my talk is probably going to end, about 10 or 15 years from this date, 10 or 15 years prior to this date. And of course, these are personal reflections. So if another person, of my age gets this talk, then probably you will get a different perspective because there are different interests in human research. There are different experiences and exposures in human genetics. So these are only personal reflections. And again, like I said, that you get these reflections from another person who's been in human genetic research in India for a long time, you probably will get a different perspective. Be that as it may, there will be some commonalities, and as I was preparing this talk, it turned out, or at least it dawned on me, that this is really trying to get my slide. This is really my obeisance to those who made human genetic research possible in India. So there will be a lot of history that I will talk about, and essentially I'll be very respectful to people who actually have made possible human genetics, the conduct of human genetics in India. So I will start with the first annual conference of the Indian Society of Human Genetics. The first annual conference of the Indian Society of Human Genetics was held in 1974 from February 14th to 16th in Mumbai. Bombay, as it was then called, and the proceedings of the first conference of the Indian Society of Human Genetics was published under the editorship of L.G. Sangvi. So, and I'm one of those people, and I don't know how many more there are in the audience. I'm one of those persons who actually was a participant of the Indian Society of Human Genetics' first annual conference. There was a preparatory conference in 1973 that was attended only by a small handful of people, primarily doctors from Bombay area and also some people from Calcutta and other places. But that was a preparatory meeting, and the formal launch was in 1974 when the first conference was actually held. In this conference, I presented my first publication. My first publication, scientific publication, was presented in this particular conference, and this is my first publication in science, and it's on hypertritosis of the year. review of its mode of inheritance, further data, and comments. So that was the paper that I published. What was I doing in 1974? I was still an undergraduate student at the Indian Statistical Institute, but already initiated into human genetics and already passionate about human genetics. So this was the first paper that I actually wrote and published. Many of the youngsters may not even know what hypertrichosis of the year is, and so what I'm going to do is to show you a picture. That's hypertrichosis of the year, the hairy ears. The ear rim is full of hair. This is actually from a paper, image from a paper that I will talk about in a second. But if you can't see the hairy ears very well, the pinna of the ears are full of hair, and that's the hairy ear. So that's called hypertrichosis of the ear rims or hypertrichosis of the ears. There are families, and this is one. These are two families. These are two families collected by someone whose name I will take right now. So these are two families, and as you can see, it's only the male members in the family who are afflicted with this morphological condition, the hairy loss of hair on their ears. And you will also see that there are males in the families who do not have this trait. And so the inheritance is kind of peculiar. It is restricted to males, so one would imagine that this is Y-linked inheritance. But also there are males who are not afflicted with, do not possess this trait, and so one would imagine that the penetrance of the gene, the Y-linked gene, is not complete, so there is incomplete penetrance. You may or may not know the name of this person. His name is Kurt Stern, worked in California, and he actually wrote the first book on human genetics called Principles of Human Genetics, and it was published on January 1st of January 1949. So that's the first book on human genetics. Kurt Stern visited India, in particular visited Calcutta, and worked with a few people. And worked with some people in the city of Calcutta, S.S. Sarkar, K.R. Banerjee, and Partha, the Department of Anthropology, Calcutta University. And Kurt Stern himself was at the University of California in Berkeley. So this is the paper that they wrote and they published in the American Journal of Human Genetics. So American Journal of Human Genetics would publish these kinds of papers that dealt with traits such as hypertrichosis of the ear rims. And essentially then at that time, human genetics was mostly about morphological traits. A lot of work in human genetics during the 1960s, 1950s and 60s was concerned with morphological traits. And like I said, that I contributed a little bit to the hypertrichosis of the ear, especially with respect to its incomplete penetrance issues. And also at that time, I've also done other kinds of things. So this is an analysis of the facial and physical characteristics of some penetrable characteristics, again, morphological traits that we worked on. And this was a part of a 1971 Indo-Soviet research. Incidentally, as I go along, you will see that my talk is not chronologically arranged. It's arranged more by subject area, and therefore it's not completely chronologically arranged. So in 1971, the government of India and the Soviet Union, they signed a treaty in order to do research on anthropology, or I would say biological anthropology that included genetics. And so we were looking at various kinds of heritable traits, anthropometric traits and anthropological traits, morphological traits, and so data were collected at that time, and we did some statistical analysis of this data. Many of these will pertain to my own contributions to this particular area. This is not to downplay the contributions of others, but only to say that these are the kinds of things that I was involved in, and it's a personal reflection, it's a personal view of how human genetics developed in India, and in the context of the global development as well. So morphological traits was what preoccupied human geneticists at that time during the 1950s and the 60s. What do we know about hypertrichosis today? So this is a paper that was published in 2004, and it essentially rules out the possibility of Y linkage. It rules out the possibility, and the way that this was done is that, again, this was done in South India. They collected a whole bunch of people who had hairy ears and who did not have hairy ears. They looked at the Y haplogroups of these two sets of individuals and essentially came to the conclusion that there were no major differences in the frequencies of the Y haplogroups among people who had hairy ears versus who did not have hairy ears, and they came to the conclusion that it could not be Y-linked. We still don't know how hypertritosis is actually inherited or what the molecular genetics of hypertritosis of the ear. It is still an open problem, but what the human geneticists thought for a very long time that it is Y-linked within complete resistance is kind of ruled out. The next phase in India, and again also globally, had to do with, again, morphological traits, but now more quantitative in nature. And this had to do with fingerprints, palms, and soles, essentially characteristics of your palms and soles. And this whole area is called dermatoglyphics. And if you look at your palm, you will see that there are these the loops and whorls and arches. You can take a close look and figure out that there are differences in these patterns. The arch is an arch, the whorl is like concentric circles and so on. So there are differences in loops, whorls, and arches. And you can go from the center of the loop till the periphery and count how many lines you are crossing, and that will also give you a ridge count, as this is called. You can get a ridge count. You can then use these ridge counts together in order to develop an index, which is called the pattern intensity. And all of these have a large amount of heritability. You can also look at this particular on the left-hand side. You see this entire palm, and there is a kind of an angle that's drawn. That's called the ISHG angle. The radial, ulnar, and the proximal. That defines an angle which is called the ISHG angle, and that angle also has a high heritability. So human geneticists then looked at these various kinds of morphological traits, now quantitative morphological traits, and that went on for quite a while. And Danuta Loish was somebody who actually spent a lot of time understanding the genetics of these dermatoglyphic patterns. But she was not the first to study this. The first to study possibly was possibly Galton, Francis Galton, and then subsequently Landon Penrose and Sarah Holt. They spent a lot of time looking at the inheritance of these dermatoglyphic patterns on fingers, palms, and soles. D.P. Mukherjee was somebody who did a PhD with Sarah Holt in Galton Laboratory at the University College London, and came back to India and started teaching. Well, I think he joined the Anthropological Survey and then moved to Calcutta University. Later, he had multiple jobs, but anyway, I can't remember. I knew him, of course, personally. He's no more, but I had known him very closely. So anyways, he moved jobs, so he worked in various places in India, various institutions in India, and he's the one who actually brought the genetic component, human genetic component to understanding dermatoglyphic patterns in India. And this was the legacy that he brought from Sarah Holt's mentorship at the University College London. Subsequently, there were many people who actually engaged themselves in dermatoglyphic research. Notable among them was K.C. Malhotra. Dr. Malhotra is still, is still quite active. He goes around giving talks and is not engaged directly into research, but does guide a lot of policy research, etc. Amitabh Basu is no more. Both of them were at the Indian Statistical Institute. Their students, Vijay Bhanu in Pune, Vibha Parmakar in Calcutta, and P. Mohan Reddy in Hyderabad, formerly in Calcutta and now in Hyderabad. They all contributed quite a bit to the understanding of dermatoglyphic traits. And I must say, the human genetics in India started primarily with people who were trained in anthropology. There were no departments of human genetics in India. There were very few departments. I think one of the first departments was started by Manish Ranjan Chakraborty in Voltaire Andhra University, Voltaire. I think the second department was started by Professor Generative Singh in Amritsar, Guru Nanak Dev University, and I know that Professor Generative Singh is in the audience. So there were no formal training programs in human genetics, and therefore anthropologists took it upon themselves to do human genetics research and also train people and initiate activities, training activities and teaching activities in human genetics in India. I have also contributed to this area of dermatoglyphics. Again, my growing up in India, and I spent most of my time on studying, human genetics in India. I actually grew up with the kind of tradition, with the kind of emphasis that was going on in India, and that was really a whiff of what was going on globally as well. So we looked at these various kinds of dermatoglyphic traits and asked various kinds of genetic questions. And most of the early papers, as you will see, were published in physical anthropology and physical anthropology or a joint journal of physical anthropology and human genetics. There are people like Jim Neal, who were, of course, cardboarding human genetics geneticists, our fathers in human genetics in some ways. Neal and Charles' book, Human Heredity, was the second book that had to do with human genetics, almost explicitly or exclusively, that was published in 1954. And Jim Neal also studied dermatoglyphics, but at that time he was looking at associations of dermatoglyphic characteristics with other morphological characteristics such as physique. There was a lot of interest in associating dermatoglyphic characteristics with IQ, and a whole lot of papers were published in associating various kinds of dermatoglyphic features with intelligence and so on. The next phase continued in India and throughout the world with quantitative measurements. And looking at morphological traits, they became more quantitative in terms of body dimensions, and anthropometric measurements were being looked at. And anthropometric measurements were being looked at primarily from the point of view of population differences, inter-individual differences, but also at the same time looking at heritability and how these anthropometric traits, quantitative anthropometric traits, were inherited. This was an early paper, early book, I must say, done by or authored by Iravati Karve and V.M. Dandekar. V.M. Dandekar is a statistician and an economist. Iravati Karve is a sociologist, a historian, more a sociologist than a historian, and I will have a few more things to say about her. But she and Dandekar did one of the first population surveys in Maharashtra, going around measuring people's anthropometric features, and they produced this book called Anthropometric Measurements of Maharashtra. So that essentially brought to the fore the fact that there is a lot of inter-individual variation in anthropometric in body dimensions, even within a small region such as Maharashtra, but across various kinds of ethnic groups, castes, tribes, and so on and so forth. So it actually brought to the fore the fact that these anthropometric measurements actually have a lot of capability. Incidentally, talking about anthropometric measurements, I must say that a lot of genetics, a lot of quantitative genetics actually arose from a measurement of body dimensions, and it started with at the turn of the century, the turn of the 20th century, 1900s. It started with Carl Pearson, who was also the founding father of statistics. So statistics actually started with a lot of statistics actually started with Carl Pearson, who was completely sold on these anthropometric measurements and how they are inherited from one generation to another, and a lot of statistical methods developed. General purpose statistical methods developed as a result of Carl Pearson's research on anthropometry, and I'll again have something to say on Carl Pearson. So the next phase that we see was something quite interesting and something quite seminal in the development of population studies in India. And at this time, population studies were confined to anthropometric characteristics, but also later flowered into other kinds of traits that humans possess. And I would attribute the first set of major anthropometric surveys first to Mahalanobis, as you will see from the dates, and then followed by Iravati Karve, and B.N. Tandekar. Mahalanobis actually set up the Indian Statistical Institute, and he was a firm believer that scientific and rational views lay the foundations of modern age, and as a result of that, he was completely sold on quantification and logical reasoning in order to infer about various processes, not just human processes, but also various other processes such as water flow and whatever else there was. Interestingly enough, Mahalanobis actually went to do a PhD at King's College in Cambridge and did not complete a PhD and came back from King's College in Cambridge because the war intervened and took up a job at the Calcutta University. The head of the Department of Philosophy at that time in Calcutta University was Acharya Brajendranath Seal, somebody who was a polymath, somebody who was extremely erudite and well-read. I had discovered at some point of time a note that Brajendranath Seal wrote to Consortium Mahalanobis by digging into Mahalanobis' archives, and I found this note where Brajendranath Seal says that Prashanto, you have to do work in India similar to what Carl Pearson had done in England. So Carl Pearson at that time was actually looking at, and this is dated 17th April 1945. It's about 30 years, 30 or 40 years, maybe 35 years after Carl Pearson had initiated his studies, looking at these body dimensions and formative characteristics among people in England, Wales, and mostly England and Wales. Mahalanobis then started to do these kinds of work, following having obtained the impetus from Brajendranath Seal, and this was the first major anthropometric survey that was undertaken in India. They called the Anthropometric Survey of the United Provinces, and this was initiated in 1941. It was initiated as a result of a collaboration between two statisticians, P.C. Mahalanobis, the father of statistical science in India, C.R. Rao, who reached his 100 years last year and is still alive, lives in the United States with his daughter, but one of the most prominent statisticians the world has ever produced, and D.N. Majumder, who is an anthropologist. So the three of them actually collaborated in this anthropometric survey of the United Provinces in 1941. I will tell you a little bit about this particular study, especially in terms of the questions that they asked. So anyway, what I'm actually showing you in this sort of hand-drawn figure, which is again from their paper published in Calcutta, which is the Indian Journal of Statistics, that was started by Mahalanobis. This is a forerunner to the principal components analysis that we use regularly. So they measured on each individual a large number of body dimensions, and these individuals were drawn from various populations of the United Provinces, primarily Uttar Pradesh, but also parts of Uttarakhand and so on and so forth. So that was called United Provinces at that time. So they took about 20 different groups resident in the United Provinces and sampled a large number of individuals from each of these groups, and this was kind of the summarization of the entire data in the form of a principal component, not really principal component, but a forerunner of it. So essentially, this is what they did. They took tribal groups, artisan groups, Brahmin groups, and then what they called as hybrid groups. So, for example, Shaktis, Muslims, and Nagaryas, they considered as hybrid groups between Brahmins and artisans, and the Chamars as between artisan groups and tribal groups, etc. And then there were a whole set of groups that they could not actually classify in terms of the hierarchy or in terms of admixture. But it's very interesting that what they were looking for is some kind of a semblance, again as a result of inheritance, some kind of a semblance among societal hierarchy versus their body semblance. And they came to the conclusion that among the groups forming the main sequence, there is close correlation between close correspondence between social status and resemblance to the Brahmins. So the closer you were to the Brahmins, there was some kind of a correspondence in your body dimensions also. However, they also pointed out that there are a number of cases in which the physical evidence definitely indicates possible origins contrary to accepted opinion. So sociologists had some opinion about the various caste groups or various population groups and body dimensions did not correspond to those expectations that sociologists and historians had. This population survey, this informative survey, also led to the formulation of Mahalanobis B squared, the B squared statistics. The derivation of the properties and applications are undoubtedly the most profound contributions of Mahalanobis, and the least squared statistic is now used in all branches of science. The next survey that was undertaken was a survey that was undertaken in undivided Bengal. At that time, East Bengal and West Bengal together, undivided Bengal. This was in 1945. This is called the Bengal Anthropometric Survey, and had a similar name, a statistical study. And we will notably that Mahalanobis has taken a back seat. He is no longer an author on this paper. It is now authored by D.N. Majumder and C.R. Rao. Mahalanobis only wrote a very long foreword to this particular publication that was published as two parts of a particular volume in Sankhya. One of the important contributions is the demonstration that regional differences within a social group, that between individuals adopting the same caste, tribal, or group label, but living in different areas. So there is, you could be a Brahmin and live in two different locations in Bengal, but you would be very fairly distinct, and individual groups that are in close proximity, geographical proximity, they show similarities in anthropometric measurements in body dimensions. So then they pointed out that a term like Brahmins of Bengal has to be used with some caution, because Brahmins of one part of Bengal were very dissimilar from Brahmins of another part of Bengal, but Brahmins of one part of Bengal were probably similar to, for example, the middle caste groups of that region of Bengal. So this was also another quite similar to what was also concluded with respect to some population groups in the United Provinces. A similar conclusion was also drawn from Bengal. Another interesting feature is that sometimes there is closer resemblance between castes within a district than between individuals of the same same caste group belong to different districts. So geographical proximity seems to have played a greater role than your caste affiliation. Geographical proximity between individuals seems to play a greater role in defining correspondence or resemblance in body dimensions compared to the population group to which they belong. Well, they actually became quite disillusioned at the end of these two major studies, the study of the United Provinces, population groups of the United Provinces, and population groups of Bengal. And they were quite unhappy with the results because they were equivocal. They did not get unequivocal results, and so they were kind of, the authors were kind of unhappy. And Mahalanobis went on to say in the Bengal Anthropometric Survey foreword, anthropometry alone cannot tell the tale. So the tale that we are looking for is resemblance vis-a-vis body dimensions or vis-a-vis biological characteristics. And then he went on to say that we have to supplement by other physical, genetic, and serological data. So this was the fourth side of Mahalanobis, that anthropometric measurements are not completely heritable, are not so much genetic compared to other genetic data and blood group data, serological data such as blood markers. The anthropologist Majumder, D.N. Majumder, says that our experience with anthropometry has not been convincing, and blood group variations also need to be looked into. So at the end of the Bengal Anthropometric Survey, they did some blood grouping, APO blood grouping, of a small number of individuals that they had recruited, and this again another way of projecting three-dimensional data on two dimensions. Three-dimensional data because we have the three allele frequencies A, B, and O, and this is a projection on the two-dimensional plane. So this is again very similar rudimentary principal components that's being done. And they also did not come, they were not happy with one single blood group system, and then they said that until we have reinforced our findings by data of other systems, such as the MNS and RH, we do not think the picture of blood groups based on the ADO system can give any clue to the ethnic origin of caste in Bengal, or for that matter in any other part of India. So this is the anthropologist D.N. Majumder. So here, now what we are seeing is that there is a moving away from such traits that that can be genetic but are also largely influenced by the environment, including nutrition, such as formative traits, and there is greater emphasis on more hereditary characteristics such as the blood groups. Also, the other realization that's coming about is that one blood group system or one genetic system does not suffice. You need a plethora, you need a battery or a battalion of genetic systems in order to in order to understand ethnic origins of castes and tribal groups of the world. So anyway, I've said this before. At about the same time, using these anthropometric measurements, there was another kind of activity that was happening, and that was to essentially place or identify hierarchies among the populations based on body dimensions, skin color, morphological traits, and body dimensions. And so there was a lot of work that was going on simultaneously among the and being spearheaded by the anthropologist B.S. Guha, S.S. Sarkar, whose name I had mentioned earlier. S.S. Sarkar collaborated with Gert Stern on the hypertritosis work. S.S. Sarkar was with the Calcutta University, so they were making these racial classifications of India. Again, a word that was not a taboo at that time. Today, if you tried racial classification, you would be hounded out. But at that time, anthropometric measurements and morphological characteristics were also being used for these kinds of classification books. I spoke about Iravati Karve, and Iravati Karve, like I said, was a sociologist and a historian, and she was working on various kinds of social phenomena in India. And one of the major social phenomena that she worked on was kinship. Kinship, of course, has to do with relationships among individuals, within families, within groups, etc. And she was actually characterizing these various kinds of kinships and also the kind of marriages that take place within groups, etc. This whole thing is called kinship organization, and she was looking at kinship organization in India. Kinship organization also has a lot to do with human genetics, and so there were many people who took cues from this kinship organization and went on to do other kinds of things, but other kinds of more genetic things. But one of the points that I would make is that this is a very seminal paper in the annals of Indian biological anthropology that verges on human genetics. This was authored by Iravati Karve and K.C. Malhotra. I've already taken Dr. Malhotra's name in respect of the dermatoglyphic work that he had done, sustained work on dermatoglyphic inheritance that he had done. It was published in 1968 in a journal called Current Anthropology. What I really, and that's the title of the paper, what I really want to emphasize is the kind of questions that they were asking. And the reason why I'm emphasizing these questions is that even at that time, many of the questions are very similar, are absolutely sharp in their content, in understanding human society using various kinds of biological characters. So this is the hypothesis that they were trying to understand, that some Indian groups formerly called, so they were looking at origins of these castes and subcastes. Essentially, the subcastes, meaning that you have, the connotation was that they had a common origin. And what they found by looking at these 8 endogamous groups of the same rank, meaning of the same social rank, and they found so much of difference in biological, there was so much of biological dissimilarities among endogamous groups of the same rank. When they set out to understand whether subcastes could always have a common origin, and they came to the conclusion that they do not, primarily because of the huge amount of biological differences that they saw in these endogamous groups. And they said that although the data from one biological survey cannot positively confirm a sociological hypothesis, such as the one that they asked, which is that subcastes would always have a common origin, it is significant that there is nothing in the data that would contradict that either. So again, it was not something that was unbelievable. It was wishy-washy, but in any case, they Maharashtra, they came to the conclusion that it's not always true that the subcaste would have to have a common origin. They have different origins as well. One of the citadels of formation of these blood group studies was the Bombay Pune area. H.M. Bhatia, who worked as a serologist, who worked at the Indian hospital in Mumbai, did a lot of work on various kinds of blood groups, and as a matter of fact, and as a matter of fact, he discovered a new blood group called the Bombay type, which is really a weaker variant of antigens, and the weaker variant essentially meant that if you tested their blood group, you would misclassify them to the O blood group and not as A or B because of these antigens. Anyway, he discovered a new group, and the Indian hospital later, and correct me if I'm wrong, anyone knows this, so if you're the hospital, that department that Professor Partha headed, eventually became the Institute of Immunology, which is now a separate institute of the Indian Council of Medical Research. It was the seeds were in AEM Hospital, and the seeds were sold by AEM with a few other people who were with, and he had a very large team of people who worked. Again, at that time, so I also moved on from informatory to looking at various kinds of genetic polymorphisms, groups, and other kinds of things, more biological in nature. So as you can see that human genetics was moving from these traits that are morphological to anthropometric, much of these traits are impacted on by environment, including nutrition, to more and more purely genetic traits, so to say, such as blood groups and other protein and enzyme polymorphisms. One of the persons that to whom we should pay our respect is L.D. Sangvi. If my memory serves me right, L.D. Sangvi was the first president of the Indian Society of Human Genetics. He certainly was the first president of the first annual conference of the Indian Society of Human Genetics. This is what I had written a very small piece on him. I'm not going to read this at all. So in Calcutta, we have what's called the Calcutta Consortium of Human Genetics. And the Calcutta Consortium publishes a newsletter called Linkage. And very recently, I had written about 4 people. One of them was L.D. Sangvi. L.D. Sangvi, like P.C. Mahalanobis, gave to the world the first measure of distance based on quantitative characters called d². L.D. Sangvi gave to the world the first measure. Well, I would not attribute the first measure. It was probably the second measure. The first measure was given by Anil Bhattacharya in Calcutta University, but it wasn't applicable to, it wasn't applied to genetics per se. But independently, L.D. Sangvi, who was doing his PhD at Columbia University, came up with a measure for measuring genetic distance based on such phenotypes as blood group phenotypes, etc. Genetic phenotypes, blood group phenotypes included. And he came up with this genetic distance measure called d². So he was one of the world leaders at that time. He came back to India after his PhD and joined the Tata Memorial Center in Mumbai, and actually spent his whole life in the Tata Memorial Center in Mumbai, and spearheaded human genetics in India. And so we definitely need to pay our respect to Professor L.D. Sangvi. As we are moving more and more into biological characteristics, L.D. Sangvi, V. Balakrishnan, and Hidalgo Kage undertook a major survey among the people of Tamil Nadu, and this was based on primarily biological characteristics. If you have not noted, please note that this was under the auspices of the Indian Society of Human Genetics, joint auspices of the Indian Society of Human Genetics and Indian Anthropological Society. So the Indian Society of Human Genetics actually spearheaded this particular survey of the people of Tamil Nadu. that led to the publication of this book for Biology of the People of Tamil Nadu. It was published by the Indian Society of Human Genetics in Calcutta at that time, and Indian Anthropological in Pune at that time, and Indian Anthropological Society in Calcutta at that time, headquartered in Calcutta at that time. The next phase, this phase continued. The next phase was a major survey that was undertaken by the Anthropological Survey of India, and this was called the All India Bioanthropological Survey. And they did this work, but a lot of statistical data that they generated, they just remained there without any analysis. And so, when I had finished my PhD with the Indian Statistical Institute, I was actually in close touch with the Anthropological Survey of India, which is headquartered in Calcutta. And one of the scientists there came and asked me if I could help with the analysis. And of course, this was the paper that we wrote, again based on the anthropometric data that were collected during the All India Bioanthropological Survey called the IBUS survey. And the data are still not appropriately and adequately enough analyzed, I would say. The data, I hope, are still remaining, and that was the time when they were noting down with paper and pencil all of the data, and so I don't know if the data are still available. The next phase was major, again, an all India level survey that was done and spearheaded again by the Anthropological Survey of India, and this study was called the People of India study. The previous one was called the All India Bioanthropological Survey study, and this was called the People of India study. At that time, it was spearheaded by K.S. Singh, who was the Director General of the Anthropological Survey of India. The People of India actually covered every one of the states of India and published independently for every state of India volumes. And this is, for example, Punjab, Arunachal Pradesh, Andaman and Nicobar Islands, and you have similar volumes for every state of India, essentially showing the characteristics of body morphology and also some social dimensions of the people of various states of India. I do want to spend, I said I was going to talk about Carl Pearson, and I do want to spend a couple of minutes, maybe 5 minutes on Carl Pearson and the aftermath of Carl Pearson, primarily because two people who were instrumental in the development of human genetics and genetics in general in the world also spent a lot of time in India and were moving spirits or moving forces behind startup human genetics, or some aspects of human genetics in India. The rediscovery of Mendel around the turn of the 20th century, in 1900, Mendel's laws were rediscovered, but it did not mean an easy acceptance of Mendel's, and primarily because at that time it was work was being done on the study of heredity on biometry. Biometry meaning these morphological characteristics such as anthropometric characteristics and body dimensions. These studies were spearheaded by Carl Pearson, and Carl Pearson and Mendel. It involved statistical analysis of mainly continuous variation found in natural populations. In humans, it pertained to anthropometric characteristics, and Carl Pearson could never reckon how discrete Mendelian characteristics, discrete Mendelian genes could actually explain continuous variation. So he was never at ease because he could not understand, or he could not come up with a framework by which continuous variation can be explained by discrete characteristics such as genes. So, like I said, Mendel studied discrete traits such as seed shape or flower color and so on, but Mendel did not study continuous variation. And when he studied continuous variation, he actually discretized those, such as heights of plants, and he called them as tall and dwarf. Did not actually deal with the actual height of the plant. So he discretized those continuous traits and dealt with discrete traits. So Carl Pearson could never reconcile how this could be continuous variation can be explained by discrete Mendelian characteristics. So again, Pearson said that Darwinian gradualism. So it's a gradual shift from one kind of measurements to another kind of measurements through the action of natural selection that could not be explained by Darwinian discrete genes. Mendelians opposed so there was another school spearheaded by William Bateson and later joined by Conniff, who actually said that Mendel was right and the biometrician's gradualism was not the right thing to think about. It's the Mendelian inheritance that was at the root of all of this. So there was this constant battle between the Mendelian school and the biometrical school. I will skip some of this and come to the year 1916. The year 1916 saw a huge battle. Well, battle meaning not fist-to-fist fight, but papers and counter-papers, publications and counter-publications happening in the pages of the journal called Biometrica at that time. And so this was 1916. Fisher came into Ronald Fisher, who was certainly a founding father of statistics. Fisher got interested in this controversy, and he studied this in his own way at depth, and came up with a blockbuster paper. And the blockbuster paper is a 34-page paper that was published in the Transactions of the Royal Society of Edinburgh, primarily because it was rejected by other journals such as Biometrica, Journal of the Royal Society. So it was rejected. Finally, it found its way into Transactions of the Royal Society of Edinburgh. It's a long paper. It's a 34-page paper, and it was published in 1918. The title of the paper is called Correlation Between Relatives on the Supposition of Mendelian Inheritance. So essentially, he's looking at correlation and not association. Association is measured between discrete traits. Correlation is measured between continuous traits. So he was able to explain correlation of relatives, correlation of body dimensions of relatives, 2 generations apart or a generation apart, based on Mendelian inheritance. And I'm of course not going to explain how he was able to do it, but essentially what Fisher showed was that biometry and Mendelian were compatible. So this actually laid to rest this battle between the biometrical school and the Mendelian school, and that ushered in a major breakthrough in understanding genetics, not just of humans but also other animals and others. So this is Ronald Fisher, a pipe smoker, smoke pipe, and the reason why I'm bringing this up is first of all tell you a little bit of the history of how our science developed, our science of genetics, our science of human genetics developed, but more importantly to inform you that Fisher came to India 7 times during the latter part of his life, into the Indian Statistical Institute, visited the Indian Statistical Institute 7 times and spent quite a lot of time, sometimes in several months, setting up or helping set up the Indian Statistical Institute, helping set up the statistical structure of India, not just of the Indian Statistical Institute, but also of India, and so on and so forth. The other person, and I will just mention one more thing about Fisher in a minute, and the other thing was that this fantastic famously figure, the picture that you see in front of you, is Prashant Chandra Mahalanobis, about whom I have mentioned, and the other person, the bald-headed person, is J.B.S. Haldane. J.B.S. Haldane worked in England. J.B.S. Haldane and Ronald Fisher could never get along well together, but nevertheless, they worked in England, and J.B.S. Haldane was a communist, and at some point of time got fed up with the way England was being governed, and decided that he would migrate to India, and he migrated to India and joined as a faculty member of the Indian Statistical Institute. And he was, of course, instrumental in setting up various training programs in the institute, but more importantly, also did organize a lot of research on human genetics in particular and genetics in general. His wife Helen Spurway was again a geneticist, but she didn't work on humans. Haldane certainly worked on humans. Haldane and Fisher and Sewell Wright were the 3 people who founded the science of population genetics, and this is a series of papers that Haldane wrote, a mathematical theory of national and artificial connections. It's a series of papers that he wrote in the Transactions of the Cambridge Philosophical Society that founded, was 1/3 of the founding of population genetics. The remaining 2/3 were by Fisher and Sewell Wright. Like One slide is missing, but never mind. Or it comes later. I probably got it. But anyway, Fisher and Haldane were quite instrumental in giving an impetus, giving a push through the Indian Statistical Institute to the development of genetics and human genetics in India. Like I said, that this is all population genetics that we're looking at, and now we're looking at more and more, relationships among, or in general, origins of various population groups, and we're looking at larger battery of genetic markers and larger and only genetic markers. So this was a study that was undertaken by what's called the Indian Genome Variation Consortium, that was spearheaded by Samir Brahmachari. And it was primarily 6 CSIR institutions and the Indian Statistical Institute. I was closely involved with this study. Essentially, again, the study was to look at the relationships, genetic relationships among various groups of India. Those are the places, groups and places, as you can see, India was covered quite well, a large number of groups. And essentially, what we were able to figure out is that these groups, irrespective of where they were, they essentially group themselves by language and not by geography. But there is also, I must also, having said this, I must also say that there is a confounding between language and geography that I'll come to in a minute. This was, of course, followed by some famous studies by Laji Singh and Dangaraj, together with a group from the Broad Institute. They published this paper in Nature and essentially came up with a model for Indian populations, and they said that there were 2 major population groups of 2 major contributions to the populations of India. One of which they called as the ancestral North Indians, the other they called as the ancestral South Indians, and essentially said that all the population groups were differential admixtures between these 2 ancestral population groups. So this is the model that they came up with. We did a more dense sampling of Indian populations, and we essentially were able to show that there are 4 sort of genetic signatures or admixtures that are required in order to explain the populations of mainland India, and these genetic signatures pertain to the 4 different linguistic groups that we have in the European, Dravidian, Austroasiatic, and Tibeto-Burman. I said that this language and geography in India is kind of confounded, as you will notice that in the, or as you may know, that in the European languages are predominantly spoken in the north, Dravidian languages in the south, Austroasiatic languages are a fractured family of languages in central and eastern India, and Tibeto-Burman languages are spoken in the northeast. So these population groups have differential admixture. Incidentally, for those of you who are not conversant with this particular kind of graph, essentially on the x-axis we have a very large number of individuals who are drawn from individuals who are drawn from different groups, and these 4 different colors tell us what the genetic characteristics of their genomes are, and these genomes can be painted depending on what the signatures are. And as you can see that there is some amount of admixture. So, for example, there are Austroasiatics who have signatures of the Dravidians, who have signatures of the Europeans and Tibeto-Burmans, and their own, which is the sky blue, which is the Austroasiatic. So, essentially, there are differential admixtures among individuals who claim themselves to be of different groups. We published this paper in PNAS. Like I said, that these studies on kinship organization, etc., also has a lot of indication on how diseases can be seen in populations, and these diseases can be seen in genetic diseases, and these diseases are a result of kinship organization and a result of the mating practices within these populations. So inbreeding is a phenomenon that's very rampant in southern India, also among the Muslims all over India. So and the Parsis. So inbreeding is a phenomenon that leads to homozygosity, increased homozygosity. Increased homozygosity leads to an increase in prevalence of recessive disorder. And inbreeding and human population studies were conducted by, all 3 of them are students of J.V.S. Haldane, P. Meera Khan, who eventually migrated to Europe, P.R. Raju, who eventually migrated to USA, A.K. Roy, who eventually migrated to Canada after Haldane's death. But all of them, when they were in India, when they were students of various Haldane, did reading studies and came to certain conclusions that I will narrate in a moment. The other person who actually spearheaded a lot of theoretical population genetics work under the mentorship of various Haldane at the ISI was Suresh Jayakar, who actually moved away to Italy after various Haldane's death. So again, I'm not going to be complete in any way, but Sanil Basu, who was at the National Institute of Health and National Institute of Health and Family Welfare in Delhi, did a large number of studies in the North India on effects of consanguinity, and the expectation for all of these studies was that we will find a higher incidence or a higher prevalence of recessive disorders. Another person who later did a lot of studies in South India was Appaji Rao, primarily with Alan Bittles, Appaji Rao, Rana Ramadevi, and Savitri. All three of them were at the Indian Institute of Science in the Department of Biochemistry, and Alan Bittles was from Australia. There was a very useful collaboration for many years, and that led to a lot of studies on consanguinity. And this is the chapter in this book that was actually edited by Dibal Subramanian and Appaji Rao for the Indian Human Heritage. Also, Alan Bittles and Appaji Rao wrote this seminal paper in Science in 1991. What were they looking for? They were, like I said, that the level of consanguinity is fairly large, fairly high. In various population groups of India. For example, it's widely prevalent even among Hindus of southern India, Muslims all over India, and also Christians and Parsis. While in the West, the proportion of marriages is only consanguineous mating between relatives is only about 1%, while in India it varies anywhere between 20 and 30%, depending on which part of India or which community you're looking at. Interestingly enough, and this is from the paper from the seminal papers published by Alan Bittles and Appaji Rao in Science, where they looked at prevalence of inherited amino acid disorders, and these are recessive amino acid disorders, and they were studying inbred groups in Karnataka. And what they found was that the prevalence of inherited recessive amino acid disorders was of the same magnitude as the outbred population. This was contrary to expectation, because what you expect is that the prevalence of recessive disorders will be higher in inbred populations compared to outbred populations. But they did not see this. Now, what was the explanation? The explanation was actually given by L.D. Sanghvi. Whether that's the correct explanation, we still don't know, but that's certainly a plausible explanation. Was given by L.D. Sanghvi a very long time ago, when there was again a lot of controversy, a lot of argumentation that was going on regarding genetic load in human populations. What Sanghvi had argued is that in these populations that practice inbreeding, we don't see a higher prevalence of recessive disorders, primarily because these recessive disorders are selected against, and over a period of time, over a long period of inbreeding, the genes that are responsible for these recessive disorders have become very small in the population, have reduced in frequency in the population because of natural selection. So there is a counteracting balance between inbreeding and natural selection, that negative natural selection that happens, as a result of which we do not find a higher prevalence of recessive disorders in populations of India. So this was the argument that L.D. Sanghvi gave, and this argument was given during that genetic load debate, as also in the biology of the people of Tamil Nadu. It's played quite well. So I'm not going to go through this. More recently, actually, P.S. Sundarrao preceded Appaji Rao's work. So anyway, Sundarrao, L.D. Sanghvi, all of these people actually contributed to these kinds of studies on inbreeding in India that results from the mating practice, marriage practices prevalent in some of the populations of India, and what they were able to relate the lack of excess homozygosity or excess prevalence, excess prevalence of recessive disorders to negative natural selection. I will sort of coming to an end of my talk. I want to end with sort of application of or development of medical genetics in India, of which I don't have a very firm grip, but I do know this, that this doctor by name G.S. Mutalik, whom I had met during the first annual conference of the Indian Society of Human Genetics, worked in he was a doctor, he was a clinician, worked in Western India in the Bombay-Pune region, and did a PhD. Well, I'm not very sure whether he did a PhD with Edmund Murphy or not, but Ed Murphy was one of those people who brought in medical genetics, genetic counseling, etc. into the world. But Mutalik did train with him. Again, I don't know as what, as a PhD student or what, but at the Hopkins, Johns Hopkins University Department of Medical Genetics, where Ed Murphy was at that time. Mutalik came back and essentially sowed the seeds of medical genetics in India. And medical genetics in India prospered. We had Mr. Partha, who established the Department of Genetics in BJ Medical College in Mumbai. That was again a citadel of medical genetics in the country. I.C. Verma, Ishwar Verma, who set up the medical genetics in All India Institute of Medical Sciences, and after his superannuation, set it up in Sir Gangaram Hospital. Again, there's a fantastic following of Dr. Verma right now that has developed in Gangaram Hospital, and Sham Agarwal in SGPGI Lucknow, who has set up medical genetics in the country, and he trained with Arnold Mokrowski. There are, of course, like I said, that I must be missing many other, many other institutions where medical genetics develop. Right now, medical genetics, in addition to this, is done in Dr. Jaroop Singh set up a major medical genetics component in his department in Guru Nanak Dev University, and there's a major medical department, medical genetics activity in Center for DNA Fingerprinting and Technology. So you will pardon me if I'm missing some departments or missing some institutions. It's not my intent to be comprehensive or as complete as possible, but I'm just giving you a flavor of this. I said that Ishwar Verma has left a good legacy in Sir Gangaram Hospital, and this is it's really a pride for us that Ratna Puri, who is again from Sir Gangaram Hospital, does medical genetics, has just published this paper in the New England Journal of Medicine, where they discovered a new syndrome, and this was published in the New England Journal of Medicine. Of course, it's a global collaboration. Today, it's not easy to find a new syndrome unless you have multinational collaboration, and this was multinational collaboration. But it's a matter of it's a very nice that Sir Gangaram Hospital is represented through Ratnapuri in this publication. So we are making a lot of impact even in the area of medical genetics. This is a survey paper that was or a review paper that was published some years ago. I can't find the date, but maybe 3 or 4 years ago, from Sanjay Gandhi from SGPGI Lucknow, and again Subha Partha, who is one of the senior medical geneticists of the country, has trained a large number of medical geneticists in the country, has written this paper along with a student of hers. And there are, in this paper, you find a list of novel genetic syndromes. In addition, the New England Journal paper is just days old, but this is over a period of time what Indian medical geneticists have been able to contribute various novel genetic syndromes that have been reported from India. I don't even know the characterizations of many of these syndromes. I know that Handigodu disease was actually discovered by Professor S.S. Agarwal and he did a lot of work on Huntington's syndrome. But again, we still don't know the molecular basis of Huntington's syndrome. Medical geneticists have, over a long period of time, looked at mutation profiles of various common genetic disorders and have established that mutation profiles of many of the genetic disorders are quite different in India compared to compared to those compared to the profiles of the same disorder in other parts of the world. And that has a lot of implications in terms of genetic counseling. That has a lot of implications in terms of providing genetic services or prioritizing how you're going to look at these various kinds of conditions with respect to their molecular basis. This is, I think, the last slide. I must say that subsequent to this, subsequent to all of these, there have been many other works that have been done. And I said that I'm going to sort of underplay the now part of my talk. I've only talked about the then part primarily. The now part has to do with lots of very interesting work that's being, human genetics work that's being done in India at a globally competitive level. Fantastic publications in the best of journals in the world. We wish we could do more, and certainly with more funding, we could do funding and sustained funding, we could do more. And I will also acknowledge over a period of time, what has happened is that the Department of Biotechnology, after it spun out from the Department of Science and Technology, has actually funded human genetics, development of human genetics in our country in a massive way, in a major way. And many of these studies that I have mentioned to you were actually funded by the Department of Biotechnology. There are other developments that took place subsequent to where I kind of ended. Linkage mapping of diseases. Many of us undertook linkage mapping of diseases. It was hard to do, and I don't think that there is any major impact on disease mapping using linkage from India. Again, correct me if I'm wrong. I don't think that we have made any major contribution by mapping the disease using linkage methods. We have done some. We have defined recombination fractions, etc. But I don't think a major disease has been mapped from India using linkage methods. Association studies, on the other hand, we have done quite well of various kinds of diseases, diabetes, IJIB, Gurgaon Bharadwaj's work, V. Mohan's work from Chennai, diabetes internal association studies, cardiovascular disease, liver disease. So we've looked at various kinds of diseases, and association studies have been done quite well. In more recent times, there is another set of diseases that have been looked at, which are called triplet diseases that have to do with mental health. And triplet diseases and mental health have also been looked at in some detail in various institutions in India. In more recent times, genomics of cancer is being looked at, genomics of host-pathogen interactions, infectious disease, and the role of the host in infectious disease susceptibility and severity. Genomics of that is being looked at, including responses to vaccine. So all in all, it's developed in a major way, and we are right now, because we have become very globally competitive. And like I said, that if only we could generate more funds, we could probably do more in-depth studies. The other, handicap that we have in India is our collaborate. I think we need to collaborate more in order to be able to, in order to be more impactful in our. We are joining hands with international collaboration. So a lot of cancer work has been done under the International Cancer Consortium. India has just now become a partner of the International Alliance. There is also a proposal for India to become a partner of the Human Cell Atlas. So Human Cell Atlas is primarily looking at the human body parts in organs, cells in human organs at single cell level. So which genes and which cell types are expressed at what stage of organ development during the life course of an individual, that we looked at by Human Cell Atlas. The International Common Disease Alliance essentially aims to identify functions from maps. Its subtitle is From Maps to Function with respect to human disease. So India has already become a member of the International Common Disease Alliance, and like I said, there's a proposal for India to become a partner of the Human Cell Atlas as well. There are major collaborative projects, like I mentioned the fact that we don't collaborate enough, but there are now some major collaborative efforts that are happening in India. One is the Genome India project that aims to look at overall genomic diversity, but in relation to phenotypes. So far, what's been looked at are the relationships between among the population groups, genomic relationships among the population groups, and mixtures that have happened over the evolutionary time scale, etc. But now in the Genome India project, in addition to that, they are trying to tie up the genomic backgrounds of individuals with phenotypes. There's also a One Health program that's happening that ties up human health with cattle and pathogens and so on and so forth. So it's called the One Health program. There's also a human microbiome program, the gut microbiome, the skin microbiome. The microbiome has a larger presence in the human body than the human cells themselves. So the human microbiome is now been associated with various aspects of human health and disease, and there is a major human microbiome project that's again a collaborative one that's being funded and spearheaded. It's being spearheaded by NCCS, but being funded by the. So all in all, the development over the last, I would say, from the 1960s, or if you go back to those formative population surveys from about 1940s till now, has been phenomenal, both in terms of conceptualization of how we look at humans, how we look at human populations, how we look at human groupings, and how we look at genomes, how human genomes can actually provide an understanding of all of those various phenomena that comprise the human. So it's really been very nice, and the Indian Society of Human Genetics has played a role. I mentioned about the biology of the people of Tamil Nadu. That was actually undertaken by the Indian Society of Human Genetics, joined with the Indian Anthropological Society. So we need to undertake more from the podium of the Indian Society of Human Genetics to be possible we can. We can't fund, but at least we can bring people together and have joint proposals submitted to the various funding. So all in all, it's I personally have enjoyed doing human genetics. I still do. I've not retired myself from contributing to science yet, but it's really been a great pleasure for me to have attended the first annual conference of the Indian Society of Human Genetics in 1974, and to have seen through the development and to have contributed to the development of human genetics in India. Thank you very much. I greatly appreciate your patience and your kind hearing. Thank you.
Thank you, Professor Partha, for giving a complete glimpse of history of ISHG till the recent developments in the genomic areas. And I think which was the particular talk was very, very appropriate for the Distinguished Scientist lecture because it really covers the entire historical aspects of the genetic scenario in the country. So I'm sure you must have raised a lot of excitement. If there are any queries, you can ask Professor Partha Majumder. Yeah.
I'm trying to get rid of my but thank you. Trying to get rid of my screen. Thank you. Yeah, I'm happy to take any questions.
So, are there any queries? Yeah, Professor Partha, there are some queries in the chat.
In that box, I see some queries.
So, someone.
Yeah, go ahead. Are you reading the chat box, Vinay?
Yes. So,
Okay, so why don't you read it?
yeah, so Mohammed Jasimuddin is asking you that what is the genetic basis of microcephaly and macrocephaly in developing brain, the role of ASPM gene, can be detected physically from head circumference or brain volume? I think this.
Look, I won't even dare to comment on it because I don't work in microcephaly, so I'm not even abreast with the literature, so I have no answer to your question.
Yeah, so this was not really covering the talk area. So Mustaq says talk was very informative. Vijay Kumar says, dear sir, it was very informative talk. Sir, kindly shed some light on the benefits of understanding host genetic polymorphisms in host pathogen interaction when in case of an infectious disease environment plays a major role.
Yeah, and that requires a separate talk. But certainly what we have seen, even with respect to SARS-CoV-2, that a lot of individuals have got infected with SARS-CoV-2. Some individuals have remained asymptomatic, and some individuals have had to, get admitted to the hospital. So in terms of clinical outcomes of infection, there is a huge difference, and some of this difference can be attributed to the genomes of the people who are infected themselves. So the genomic backgrounds of the infected do play a role, and there are some major studies right now going on around the world with respect to SARS-CoV-2, but with respect to many other infectious pathogens, there are already quite a number of indications at least that host genomes play a role.
Yeah, so I think in general everyone has the appreciation for the talk, and I have just one query that in olden days, also during the prior to ISHG's initiation, there had been some database on the dermatoglyphics in the Calcutta itself there, and due to which our Sir Harshal was the ICS officer. He actually was maintaining the huge database of this fingerprint parameters. So was it not accessible to the Indian Anthropological Survey of India, or was it not a part of Anthropological Survey of India collections there?
I don't think the collection was a part of the Anthropological Survey of India. No, I don't think it was it was primarily interests or private initiatives that were taken in order to collect those. So, for example, early on, some of the anthropometric work were done by the British, and they were not under done by Anthropological Survey or any such agency. They were almost privately done, private funded by the British government at that time. And we know some of these were actually published later, but where the data have gone, we have an issue.
And then there had been some, I think, issues with Fisher and Ney's experiments also. So where the Ney has actually commented on the Fisher's experiment, something of that sort.
What is the name?
M. Ney. Masatoshi Ney.
Yeah, so has commented on what?
On the, so there were some issues with the Ney and the Fisher there on some linkage studies there.
Fisher? No, but they are generations apart, so to say. No, Fisher. When Ney started contributing, Fisher was already had already receded.
Right.
No, I don't. I think you're talking about Fisher and Wright. So Wright.
No. Ney actually has talked something on the mutational basis of Fisher's observations there. So
I don't know that. I don't know that. So look it up and also share with me, please.
Yeah, sure.
Yeah.
So, and I think you had worked with Professor Ney, right?
Yes. Yeah, we have published together as well. Yes.
So I think we have two persons, you and as well as Professor Arvinda, who had actually worked with Professor Ney earlier. So
yeah, the one, yes, the one Arvinda actually did a PhD. I did not actually share this information with you. Arvinda and I, we went to undergraduate. We did our undergraduate studies together at the Indian Statistical Institute. you. So we are
Okay. So, are there any queries, anyone? Brother Jairut Singh?
Partha, that was a lovely talk. Thelma here. Can you hear me?
Yes, then I can recognize your voice. So.
Oh, okay, perfect.
Yes.
All right. So, I just want to ask you, because you've talked about then and now, would you like to tell us a little bit about your thoughts on what the next 20, 25 years will hold for us with all the new technologies? Would we reach what we are looking for or the way you visualize? That'd be very nice to hear from you.
So, Velma, you're probably better to imagine and better placed to imagine and also comment. But anyway, let me share what I think will happen. What I think will happen is that a lot of these diseases that we are dealing with right now are quite complex. There are two areas that human genetics is going to be driven. One area is to be able to identify effects of multiple genes or multiple loci on these complex characteristics. And we are still far short of understanding how the genes and environment play a role. We talk about it, but we don't do any control experiments, nor do we have any cohort studies. Cohort studies are fantastic and great for identifying the role of environment or the interaction between genes and environment, and we are not doing that seriously. In the coming years, I think that will be one area which will be come to the limelight. The second will be gene interaction or interlocus interaction. There also you work on complex diseases. I work on complex diseases, and at the end of these studies, we are actually left quite sad that we haven't actually deciphered these diseases in its entire detail. So I think that is going to go. The International Common Disease Alliance is pushing for understanding function from these maps. They've been constructing maps to association studies or linkage studies, and now looking at function. That's another domain where it will go. The last, which will be very technologically driven, very computational, both computation and the wet biology technology driven, will be the area of understanding heterogeneity. The same disease affecting the same organ display different clinical outcomes in different patients, and that will depend on the kind of cell types, both new and other kinds of cell types, that will be helpful understanding wider heterogeneities in clinical outcomes, mostly at the single cell level. That's expensive. Wetland is difficult, computation is more difficult, but we must get in there in order to be able to human genetics in the years to come. So these are the areas where I think human genetics is going to go.
What about the large proportion of the non-coding sequences? More and more of that kind of.
Yes, that also includes, like when I said that interlocus interaction, I didn't only mean coding loci, it could also be regulatory and non-coding loci. We will, so I just put it in a very broad framework without actually mentioning coding or non-coding, but yes, certainly at least regulatory polymorphisms we need to take into account to understand what we mean by incomplete penetrance or complexity of a disease. So this will be the thing that we will have to look at.
So anyone else?
My friend Majumder Singh, who is an IAS officer in Rajasthan, is asking whether I could possibly talk. She spearheads the science and technology in Rajasthan. Majumder Singh is an IAS officer, very interested in this. I've known her for some time. I've never actually met with her face to face, but known her for some time, organizing various events through the Academy of Sciences, and she had no mood to write this up. It takes a long time.
Yeah, but I would, we would like to share this particular talk in the YouTube there. So I hope you have no objection to it.
Oh, you can do that. Yeah, you can do that. I don't think I've made any mistakes, but you can do that.
Right. So, and then there is another query from Khushdev Bandesh. He's asking that so many field of in human genetics, what do you feel about how much is the contribution of environment to common lifestyle disease there?
How much is the contribution of the environment on huge contribution, of course. And I just said that one way to understand the role of environment to common diseases will be to organize cohort studies on diseases, cohorts of diseases in multiple places. Those are extensive follow-up is also a difficult problem. So that needs otherwise diseases. So I do feel that there is a lot of role of the environment.
And I think there are no more queries. There is one thing I came across your talk that you have said that the Brahmins in Bengal are actually do not have the same origin there. So so Brahmins in Bengal actually come from two different several clusters. They are not of the same origin there. Is it right?
Correct.
So
Correct.
does the modern tools? Yeah, does the modern tool actually dissect it?
It's not my conclusion, it's the conclusion from the Bengal Anthropometry Survey.
Yeah, so I'm saying that is the modern tools, does it actually further explores it further?
We actually have looked at Brahmins of Bengal, but not at the same depth as the Bengal Anthropometric Survey, meaning that we didn't actually sample Bengal from different districts of Bengal and then try and admixtures, and there are differences in admixture levels across these various places. So no, honestly, we haven't looked at that in any great detail.
Okay, so there is another query that, good afternoon, sir. You have talked about the studies from different states in India. Do association studies regarding disease susceptibility different states can be helpful there?
Different what can be helpful? Sorry.
The association studies regarding the disease susceptibility from different states, can it be helpful? Ideally, I think he means to say that. Does it actually the state-specific susceptibility studies one has to carry out?
State, I honestly don't know. Certainly, regions or in different regions or different populations, primarily because these association studies are being done for various kinds of common diseases where there is a lot of impact of the environment, and therefore to understand whether the association holds up in different environments, then it becomes a generalizable conclusion to show that we need to undertake these studies in multiple environments or in multiple population groups.
Yeah, so there is another query. Suraj is asking that is there any effort to establish Biobank in India similar to UK Biobank? I think you are a part of it, sir.
Well, there is Biobank has been set up by the Department of Biotechnology and the Regional Center for Biotechnology in Faridabad. Now, who is contributing? How it's being organized? I have actually, I was a part of the committee that in principle agreed that a biobank needs to be formed, but how it's going to be organized depends on the principal investigator and the collaborators of the biobank. I still haven't. It's only been recently that it's been approved, maybe less than 2 years ago. So it's, I think, still in the process of being set up. But whoever asked this question, I think, is a very important question, because that organization will mean, what the payoff from that investment will be. Unless it's organized well and planned very well, it's just going to be wasteful investment of expenditure.
So in the event of no more queries, I would like to thank chronic disease increasing in India. Is there a genetic role behind it? I'm sure, yes. So I would like there's some query that chronic disease is increasing in India. Is there a genetic behind it?
Yeah, of course.
Yes. So anyway, so thank you, Professor Partha Majumder, for your excellent talk, giving us a complete overview of the historical aspects of genetic scenarios in the country, and keeping us updated with the recent trends of working out with the cellular mappings and all. Professor Jairup Singh, do you have to say anything, sir? So I wish to end this session, and I would like to thank Professor Partha again that he took time off to spend some time with us and giving us an orientation of the entire perspective of the talk, and which was really very appropriate for the Distinguished Scientist Lecture Series, sir. So thank you very much once again for taking time off from your work. Thank you, sir. And thank you all for attending this talk through the various online medias. Thank you, sir.
Thank you very much for giving me this opportunity, and I'm really honored by being able to give this talk.
Thank you, sir.
Thank you.
Okay, thank you.